Good afternoon, everyone. We are done with protein chemistry. We are done with amino acid chemistry, carbohydrate chemistry, lipid chemistry. So we will today get done with protein chemistry as well. Okay. But before that, are you ready? Okay. From the amino acid chemistry. Let's see how much do you remember. You can see the books, you can read your notes. That's fine. I want answers though. Okay. Easy questions. True regarding solubility of amino acids. Soluble in non-polar solvents. Soluble in polar solvents. None of the above, all of the above. This is the physical property of amino acid. Anyone that can remember? Polar solvents में soluble था कि नहीं था? Do you have notes? Yes, how many of you say A? Okay, B? C? D? None. So you're divided halfway. Soluble in polar solvents and not soluble in non-polar solvents like benzene. Next, optical isomerism is because of asymmetric carbon. Good. Next, aromatic amino acids. By the way, which are aromatic amino acids? नाम बता दो एक. Tyrosine. Okay, good. So it shows absorption at which wavelength? 280. Okay, good. Next, the pH at which an amino acid has no net charge. Okay. Come. Which amino acid is involved in the synthesis of histamine? Easy? Histidine. Next, D amino acids, reaction with ninhydrin produces which color compound? Which color? It was bluish purple color residue. Next, carboxyl group reacts with ammonia to form C? Yes, very good. Transamination reaction is B? All of you B? Amino acid to a keto acid. So the original amino acid becomes a keto acid and the original keto acid becomes an amino acid. Which of the following is a derived amino acid? वहाँ में से last वाले में से बताओ. Question number 9 का answer. Which one of them is the derived amino acid? Which is the correct option? No idea? Second. Collagen? The rest? Okay. This one. Which is the correct option? Number 9. Next. D. All of the above? This side? Do you agree? All of them are derived amino acids. Okay, last question. Zwitterion. All of the above. No net charge, no movement in electric field. All of the above. Okay. Now we'll start proper protein chemistry. I did this MCQ so that you remember what we are talking about. Okay? Please no talk. Now I will talk and you will answer. Now, protein chemistry. Here we'll understand what protein is, which you already know. The function of protein, the classification of protein, and the structure of protein. So, proteins are just polymers of amino acids joined together, right? These balls are different amino acids joined together in this manner. This is called a peptide then, and when they are folded, they are known as protein. Okay, so this is protein. Now, there are certain terminologies that I think you should be aware of. When we say dipeptide, like I said, this is called a peptide, okay? So when there are two amino acids joined together by one peptide bond, they are known as dipeptide. Dipeptide sounds like two peptide bonds, right? But it is not two peptide bonds, it's one peptide bond joining two amino acids. So that is dipeptide, like what we see in aspartame, which is a sweetener. Aspartame is made up of aspartic acid and phenylalanine. Okay, fine. Then we have something called tripeptide, meaning three amino acids joined together by two peptide bonds, like the peptide called glutathione. Glutathione is made up of glutamic acid, cysteine and glycine. Very, very important compound. You will read about it later on, so I'm not going to details of what it does, but it is very important. Remember, it has three amino acids. Very common question in Viva. What is it? What is glutathione made up of? It is made up of glycine, glutamic acid and cysteine. It has an antioxidant property. Same way, what will be octopeptide? How many amino acids? 8 joined together by 7 peptide bonds, right? Like oxytocin and ADH, vasopressin. These are hormones. So now you know hormones are also proteins. Then we have polypeptides, meaning 10 to 50 amino acids when they are joined together. These are known as polypeptides. Example, insulin. Now, this is a polypeptide chain, alright? This is a polypeptide chain. So, whenever this polypeptide chain is there are two ends to it. One is called N terminus and one is called C terminus. Wherever the beginning is, which is starting amino acid is, the NH3 part, the amino part of that particular amino acid is known as the N terminus. Okay, this is the starting point. And the last amino acid, the carboxyl group of the last amino acid, that side is called C terminus. So everywhere you will see, now you will talk about polypeptide chains more, there will be an N terminus which is the starting point and C terminus which is the ending point. Okay. Now classification of protein is done on the basis of these 4 categories. One is based on solubility, another is based on shape, the third is based on function, and the fourth is based on composition of the proteins. We'll go through one by one. This is based on solubility. So the protein that is soluble both in salt solution and in water, they are known as albumin. They are bulky proteins. Then globulins are the ones that are soluble in salt solution but sparingly soluble in water, which means that they can dissolve in water, but they are minimal dissolution in water. Then we have something called histones, which are only soluble in water, and the other proteins that are neither soluble in water nor in salt solution are called scleroproteins. Now this is based on shape. You see 2 different kinds of shapes here. There are fibrous protein and then there is globular protein. So easy to understand. Fibrous protein are long thread-like, hair-like proteins. They have an axial length and breadth which is more than 10, which means that the length is much more than the breadth. So this is called as fibrous protein. Where do you think we will have these kind of proteins? On our hair. Okay, so example is keratin and myosin. Keratin is a protein that is present on our hair, then on our skin and our nail beds, etc. Okay, so this is globular protein. Understandable from the size, it is globe-like. Obviously it is not perfectly spherical, but compared to this, fibrous protein, this is much more round shaped, spherical shaped. This is globular protein. So example is albumin and globulin. Globular protein, globulin. Okay, these are bulky ones that I told you before. Then based on function, different functions of proteins are there. For example, structural, the proteins that give structure to our body, they are known as structural proteins like collagen, keratin, myosin. Myosin is present in the muscle fibers. Then some of the proteins act as enzymes. You must have heard 1 or 2 enzymes by now. In digestion also you must have read pepsin, amylase, lipase. All of these things, they are known as enzyme protein. Proteins are called enzymes which are used in enzymatic activity. Then some of the proteins act as hormones like insulin, glucagon. Then we have transport proteins that transport things, right? Like hemoglobin and myoglobin. What does hemoglobin transport? Come on. Oxygen, right? Myoglobin, same. Then we have storage. Ferritin is a storage form of iron. Ceruloplasmin helps in storage of copper. Okay. Then we have protective called antibodies. You have heard of antibodies and antigens, right? Especially during COVID days, you must have heard about it. So antibodies are proteins made up of proteins. Some of the proteins are classified as antibodies because of their protective property. Then finally we have something based on composition. Simple protein, which means that the protein is purely made up of protein. This particular thing is made up of protein. There is no extra material attached to it. Okay, there is no non-protein part in it, like albumin, globulin, collagen, and all of that. So what will be conjugated proteins? So this simple protein has no non-protein part. So conjugated proteins will have non-protein part. Very good. So they have a non-protein part called prosthetic group that will be covalently attached to the proper protein. So there is a combination of protein and a non-protein part together, right? And they are covalently linked, so they cannot be separated from each other. So if you separate them, then the whole protein or the compound will lose its activity. Okay, so examples are glycoproteins, lipoproteins, nucleoproteins. What are glycoproteins? Combination of carbohydrate and a protein, right? So carbohydrate is a non-protein part and protein is a protein part. Same thing with lipoproteins, combination of proteins and lipids. Then what are nucleoproteins? Nucleic acid and proteins. What are nucleic acid? Say your DNA, right? So DNA is a nucleic acid, combining it with another protein forms histones. So example is histone, that is a nucleoprotein. Okay, so that is a type of conjugated protein. Another protein that we see commonly is hemoglobin. Hemoglobin has a protein part and it has non-protein metal as well, like iron. It has a porphyrin unit and it is a heme unit. Okay, this is another example of conjugated proteins. Now we talk about structure or organization of protein. Very, very important for your exam, important for Viva. It can come as a long question, as a short question, as an MCQ, everything. You have to know everything regarding the structure or organization of protein. It is not difficult, it is very easy. Okay, so we'll go into that. The structure of protein is divided as primary structure, secondary structure, tertiary structure, and quaternary structure. Simple, right? We'll go through that one by one. First is the primary structure. Primary structure just means the sequence of amino acid in a protein. Okay, so this is the linear sequence. The word is linear sequence or order in which the amino acids are placed in a protein. Like this, these are the amino acids. If you can imagine amino acids, each ball, and then peptide bonds. This is insulin. So this is an example of primary structure of protein. This is called primary structure. Now, bonds that we see in primary structures are peptide bond and disulfide linkages or disulfide bond. We'll talk about this. This is an insulin, structure of insulin, which is an example of primary structure of protein. You would see here insulin, human insulin. You hear about diabetes all the time and insulin all the time, right? So, insulin is produced by the beta cells of which organ? Pancreas. So pancreas secretes insulin. This is the insulin. Human insulin looks like this. It has two chains, chain A and chain B, or B chain or A chain. A chain consists of 21 amino acids. B chain has 30 amino acids in them. But what I want to focus on is these. These are, what are these? Sulfur, right? Disulfide, meaning two sulfur, disulfide bond. Apart from peptide bond that we see, we also have disulfide linkages. Now these sulfur linkages, disulfide linkages or bonds are seen both intra-chain and inter-chain. Inter-chain matlab, ye do chain ke beech mein there is a disulfide bond. Then within the same chain itself also there are disulfide linkages. You will see over here, this is the starting one. So 1, 2, 3, 4, 5, 6, between 6, 7, 8, 9, 10, 11. So 6 and 11th number of the cysteine residue of the A chain of insulin has disulfide linkages. This is intra-chain, within the chain. Then between two chains also we see over here, between 19 and 21, over here 6 and 6. There is disulfide linkages. Where do we see sulfur? Which amino acids? Two amino acids have sulfur in them. One is cysteine and another one? Methionine. Very good. So these things please remember because they will ask you in Viva. Now we come to secondary structure. So when this primary linear chains folds into itself and makes a shape, that is known as the secondary structure. Okay, they can be alpha helix and then they can be beta pleated shapes. These two are the most common that we see. There are other shapes as well, but these are the most common and that's what you should know. Indian scientist Ramachandran made a significant contribution to the understanding of this secondary structure as well. This is the alpha helix. This is a type of secondary structure that is seen. See, those of you who are not interested in class, you can go out of the class. Please don't disturb me while I'm teaching. This is the alpha helix. So, does it remind you of any structure that you have seen? One, DNA structure. But DNA structure is not an alpha helix, okay? It's a double helical structure. There will be another helix, another coiling over here, two-sided, okay? Anyway, good observation. So, right off the bat, you will see that the R group of the amino acid, you remember the structure of amino acid, there is an NH group, NH2 group, then there is carboxyl group, there is an R group, right? So that R group is projecting outwards. The way that these bonds are arranged, it looks like a helical structure or a coil, like these, okay? So there is hydrogen bonds. You see there is hydrogen bond over here. This hydrogen bond is formed between the peptide bond of the, the nitrogen atom of one peptide bond and the oxygen atom of the fourth peptide bond. Okay, not 1 and 2. It makes 1 with the fourth one. That's why the shape becomes like this. This is the most common structure, secondary structure of protein. It is the most stable. It is seen in fibrous proteins like collagen, keratin, myosin. So right-handed one is more stable than the left-handed one. So if you raise your thumb, so thumb will indicate the direction in which it is going. So over here you will see this one is going upwards, okay? So the coiling is going upwards. So right-handed will be, if you put your thumb over here like this and the coiling of your fingers, this will be your right, not towards like this. Towards like this will be the direction of the coiling of the peptide bond, the polypeptide, so not peptide bond, polypeptide chain. So this is how a right-handed helix looks like. So you see there are many turns in them. Each turn will be 0.54 nanometer and it will have 3.6 amino acids in them. And each amino acid will be 0.15 nanometer separated. They will be apart from each other. There is a space of 0.15 nanometer between each amino acid. They have hydrogen bonds, like I said, between the one peptide bond and the fourth peptide bond. Except all of the peptide bonds are involved in hydrogen bonding except for the first and the last one because they are free, right? Then there are certain amino acids which are known as disruptive to alpha helices, like proline, acidic and basic amino acids and branch chain amino acids. They do not form alpha helix, okay? So can you name any branch chain amino acids? Do you have your notes, your books? You can just go through them and tell me any one branch chain amino acid. Anyone? Proline? Valine, okay. Leucine, isoleucine and valine. Okay, those are branch chain amino acids. Good. Then the other form of secondary structure is beta pleated sheets. What are they? अभी तो ये coiling हुआ. If different polypeptide chains, when they are arranged longitudinally, if we extend those polypeptide chains and they are placed longitudinally across each other, they will be looking like sheets. That is called beta pleated sheets. They look like this. So this arrow indicates it's going towards the head there, right? And this is the other side. So this is one polypeptide chain, this is another chain. They're going in the opposite direction, and the way that they are arranged in space, it looks like a sheet, okay, like paper sheet. So they are called parallel and anti-parallel. From this photo image, you can see that this is going towards this chain is going forward, this is going backward. So they are in opposite direction, so they are anti-parallel. And this arrow is indicating here, both of these two chains, different polypeptide chains going towards the same direction. So parallel beta pleated sheets. Okay, simple. Then we have something called super secondary structures and non-repetitive secondary structures. What are super secondary structures? Just the combination, the way that different types of secondary structures combine together and form one unit, that is known as super secondary structure. है वो secondary structure ही, but different types of secondary structure come together to form super secondary structure. Like here, you can see beta, alpha, beta units. This is a super secondary structure because see, beta sheets are represented by an arrow. Okay, so this arrow represents beta pleated sheets. ये beta pleated sheets हो गए. Alpha helix, then a beta pleated sheet. So this whole thing together is a super secondary structure. Then we have non-repetitive secondary structures as well. Those are just structures that are not repetitive. Like you see over here, these are repetitive structures, right? At a particular interval of time, they're the same. Same thing over here, you can see coiling is there repetitively. There are certain things that are not repetitive, they are flexible, they don't have a proper shape. They are known as non-repetitive secondary structures. They can be loops, coils, bends and turns. Okay, this is an example of a loop. So this, what does it do? It attaches, it joins the two different secondary structures or the same secondary structures. Like here, helix, loop, helix. So this is an alpha helix, then there is a loop here that joins the two chains. Then this is another polypeptide chain of alpha helix. Okay. Then we have bends and loops. Loops तो मैंने बोल दिया, bends and turns. Bends, beta bends and beta turns can be used interchangeably. They are a sequence of amino acids, usually 4 amino acids, that attaches different structures, secondary structures like these. These are bends. This is also a bend. The important thing about the bends, beta bends or beta turns, is that the first peptide bond and the last peptide bond, the 4 amino acids, there is a hydrogen bond linking these two. And this is quite strong, due to which there is a 180-degree turn. Therefore, the other beta pleated sheet becomes ulta. It reverses. So you see it's going this side, this is going that side, like these. So these are all bends. Okay, loops already told you. Then the secondary structures are joined together through the hydrogen bond, electrostatic interaction, hydrophobic interactions in the non-polar groups, and van der Waals force of interaction. Why am I telling you these? These are supposed you by heart these things because this will come in your Viva as well. Secondary structure ho gaya. Then we come to tertiary structure. Tertiary structure is the overall 3-dimensional structure of the protein. Whenever a polypeptide chain comes together, like over here, this is a tertiary structure. Isko aap dekhoge, if you microscopically, you will see that it consists of both the alpha helix and beta pleated sheets, etc. So it will have sheets, turns, bends, and helices as well. They have hydrogen bonds, electrostatic bonds, and van der Waals force of interaction. This is another structure, another example of tertiary structure of protein. Okay, so this is a 3-dimensional structure, right? We see that there are disulfide linkages. There is hydrophobic interaction. See, hydrophobic interaction means that the non-polar side chains of any amino acids, they also have interaction with each other, with other non-polar groups of the other amino acids. That is why this is known as hydrophobic interactions. They are not on the surface, they are usually below the surface. Then we have hydrogen bonds, you already know that. Then also there is presence of ionic interactions, which means that there is interaction between the positive and the negative groups that are present in each amino acids. Okay, all of these bonds help in making a tertiary structure. Then finally we have something called a quaternary structure. When two or more polypeptide chains come together, then that is form a larger unit, then that is called a quaternary structure. Simple like this. There are different colors for different chains, polypeptide chains, and when they come together, join together as one unit, then that is called as quaternary structure. Examples, hemoglobin again. Hemoglobin has 4 different subunits made up of 4 different polypeptide chains or groups, units, that is a quaternary structure. If these units are removed from each other, if you try to separate them, then the whole protein will lose its biological function. So hemoglobin may say protein part, if you remove the heme part, it will lose its function. If you remove the iron part, it will lose its function. If you remove the porphyrin part, it will lose its function as well. So to summarize, we learned that this is the primary structure, just the beads on a string. When they fold into itself and make a shape, that is secondary structure. Tertiary structure, when they form a 3D shape structure, when 2 or more polypeptide chains come together and join together and form one unit, that is quaternary structure. Now I want to talk about a little bit about denaturation. Whenever there is disruption or whenever there is disorganization within the proper original composition or the structure of a protein, that is known as denaturation. Okay, so denaturation में क्या होता है? There is disruption, there is destruction or damage to the secondary, tertiary and quaternary structure. Keep in mind the primary structure remains fine. The primary structure is not affected. This is a very, very important Viva question. Okay, primary structure does not get affected. Peptide bonds are not broken by denaturation. The other bonds are broken. There are certain characteristics of denaturation. For example, like I said, primary structure remains intact. Protein loses its biological activity though. Just because primary structure is there doesn't mean that protein will have its function. Protein doesn't have, it loses its function, it loses its biological activity even though there is primary structure remains. Okay, they become insoluble in the solvent that they were previously soluble. And viscosity therefore increases of the solution in which the protein was, surface tension decreases. There is increase in ionizable and self-hydral groups present in protein. That happens because the disulfide linkages also breaks, though sulfur ions are now free to bond. So there will be increase in ionizable and self-hydral groups of proteins. Denatured proteins are easily digested. Therefore, when we cook our food, it is easy for us to digest. The peptide bonds that are there in the primary structure or in the secondary, tertiary structures, they are available to bond with the enzymes that are present in our body so that it easily get digested. Okay. Denaturation is usually irreversible. किसी protein को denature किया तो उसको दोबारा से we cannot make it come back together. Usually, but there are certain circumstances in which we can do that. With that process is called renaturation. Example is hemoglobin. So when hemoglobin is comes in contact with salicylate, salicylate is just a type of a drug, though it undergoes denaturation. But if we remove this salicylate, the triggering factor out, then it can come back to its original position or original form, and this is seen rarely and in compounds like hemoglobin. What are the things that can cause denaturation? Heat, UV light, mechanical agitation, pressure, organic solvents, acid and alkalis, heavy metal ions, urea and detergents. Can you give me a very good example of denaturation that we see in our daily life? Boiling of egg, very good. Any other example? What is curdling of milk? Yeah, that is a form of denaturation. When we boil the milk too much, the protein, what is the name of the protein in the milk? Casein. Casein breaks down. Okay, what happens to that part? You'll see if you keep on boiling, then the solid part, the milk part, the solid part comes off, and the water part and the solid part, it separates from each other, right? So that is what it is. It becomes insoluble in the solvent that it was previously soluble. So milk has casein in it, it has water in it, right? So these two things separate from each other, it forms a coagulum. One important test that you will do in your practicals as well, this is for the test of albumin usually, heat coagulation test, where you will take a test tube, there will be a solution given to you which will be a protein solution. You don't know that it's a protein solution. So what you're going to do is, you're going to heat the upper 1/3 of it, and you will see that after some time, the protein in that solution breaks and it will form a coagulum. It will form a thick, insoluble precipitate on the upper surface, upper 1/3 of the test tube, which will show that there is presence of protein in this solution. So this is an example of denaturation that we see in real life, milk and egg. After egg, you make an omelette. Can you make it back to an egg? Boil? No, right? So this is the thing with denaturation. You cannot put it back into its previous shape after it's been broken, except for things like hemoglobin. Now, after everything that is done, now we have learned everything, whatever we needed to learn. Can you tell me what are the functions of protein? List functions of protein. What does protein do in our body? Yes, catalyzes reaction in the form of enzymes. Anything else? It acts as? It gives us immunity. Very good. Anything else? Hemoglobin? Transportation. Yes. Transportation, and anything else? Storage. Very good. What else? It gives us a structure. Anything? Okay, all of you are correct. In enzyme catalysis, and the proteins are acting as enzymes and helping reactions that happen in our body every day. Transport and storage gives us a mechanical support, gives structure to the body. Immune protection in the form of antibodies, growth and differentiation, and generation and transmission of nerve impulses. No wonder we tell that protein is very, very important for our body. Why do these bodybuilders take extra protein? You see, they have these protein powder and whatnot. They are trying to build their muscles, right? So they are trying to trigger their muscle growth. Growth and differentiation is one of the important function of protein. So this was the class. Thank you for your attention.